in 2008 at Cold Spring Harbor Lab, and this was part of a joint training program with Janelia Research Campus. And so the actual doctoral research itself was done with Carl Svoboda. And I guess Carl was moving his lab maybe between Cold Spring Harbor and Janelia at that time?
Yeah, it was three years at Cold Spring Harbor and then a year and a half at Janelia, yeah. So right in the middle, yeah.
Sounds like a great experience. And then I went on to do postdoctoral training with David Tank at Princeton University, and then took his position at Harvard in 2012. He was recently promoted to associate professor. So today we're going to be hearing about how cortical networks, from Chris, about how cortical networks guide flexible decisions during spatial navigation. And I'm very keen to hear more. Chris, take it away.
Okay, thank you, Bijan. That's great to be visiting virtually, and yeah, I look forward to the chance to present our work and hear any feedback you might have. So I will now share the slides and make sure that all looks okay. So that all looks good, okay, Bijan?
Now it does, yes.
Now it does, good. Okay, great. So and you can see my pointer, I assume, right?
I cannot see your pointer right now.
You cannot. Okay, then I will do it without pointing. Okay, great. So yeah, so my lab's interested in understanding how the mammalian cortex performs the computations necessary for complex cognitive processes, things like spatial navigation, flexible sensory motor decision making, and short-term memory. And so we use the mouse as our model subject, and we use a wide range of different methods, including behavioral methods, optogenetics, optical imaging, electrophysiology, and computational modeling. And so what I want to tell you about today are two unpublished pieces of work in which we've started to look at the cortical networks for flexible decisions during spatial navigation, focusing on kind of the functional organization of these networks and some of their causal relationships to decision making. So in the lab, we really focus a lot on the idea of learned and flexible navigation decisions, and we think that these are really fundamental aspects of natural behavior. So suppose you're walking down the street and you have to make a decision about turning left or right in order to reach your upcoming destination. You might, for example, have in one day be trying to reach your favorite restaurant, and over time you might learn that you can use things like different landmarks and street signs, these sensory cues, in order to guide your decision to walk and turn to the right in order to reach that destination. However, on another day, you might actually be trying to go to the local market to buy a piece of fruit. And so you could actually use exactly the same sensory cues to make exactly the opposite behavioral choice, in this case, to go to the left in order to reach that destination. And so we think that these learned and flexible navigation decisions are fundamental aspects of natural behavior for us, but they're also fundamental aspects of natural behavior for our subjects, mice. And moreover, we think they're things that could actually be studied within a laboratory setting. So when we think about navigation decisions, it might be tempting to start to think about them in kind of a feed-forward serial processing type of way. The idea that maybe there are sensory stimuli that can be used to inform navigational goals, where we might want to make decisions, and then translate those decisions into navigational trajectories like sequential locomotor plans and actions. I think this is really, really valuable to think of it in this way, but importantly, navigation requires also continuous feedback loops and the integration of multiple features. I'll just give you a couple examples. When we start to think about feedback loops, then the ideas of the movements that one generates as one is navigating, this can obviously be fed back in a fast way for sensory motor feedback loops, where one has to kind of continually interpret sensory cues in the context of one's self-motion. And furthermore, over longer timescales, the outcomes of certain trajectories can be used to update goals for future decision making. In addition, a lot of processes need to be integrated. These don't kind of live on their own. As I've already mentioned, sensory cues have to be interpreted in the context of self-motion. As one is forming a decision or forming a navigational goal, one has to take into mind things like the context, sensory information, position and environment, ongoing movement, past outcomes, and so on. And when one forms a trajectory to reach that goal, one has to have in mind the goal and also one's current position in the environment. So I want to emphasize that we really think of navigation decisions as quite interesting and important for kind of incorporating these types of feedback and integration type features. And we think those are thus really interesting to study for maybe more naturalistic types of behaviors. So obviously such a complex behavior will involve a wide network of cortical and subcortical areas. We think subcortical areas play a critical role in a lot of these behaviors, but for the most part, we've studied cortical areas. And so the focus of my talk today will be on the functions of different cortical areas. And so when we start to think about these different cortical areas, there's many areas you think that could be playing a role. For example, we studied a lot the posterior parietal cortex. We've shown that this area can form navigational sequences, has information about upcoming actions, and we and others have shown that there can be movement and postural information that's present in PPC that could be important for these types of behaviors. We can think about other areas like visual cortex, which is obviously important for sensory perception, and has also recently been shown by the Carandini lab to have things that look like place cells and have spatial information. Obviously, higher visual areas could be important for complex visual processing, things like landmark recognition. Retrosplenial cortex is a really interesting area that's been shown to have place cells, head direction cells, and is thought to form kind of this combination of ego and allocentric coordinates, world-centered and body-centered coordinates. Then, of course, frontal areas for planning and reporting action. So these are just a list of some of the areas that could be important. It's not an exhaustive list, but what it's trying to point out is that many areas are involved, and actually most of these areas have not been studied kind of simultaneously within the same behavioral task and the same setting. Often we study these areas kind of one by one in different settings and then try to piece together the types of patterns that we see across recording conditions, across labs, across behavioral tasks, and so on. And I would argue that a major gap is that we don't actually understand how all these areas might interact together within kind of the same behavioral context. And moreover, most of these areas have actually not been studied during decision tasks during spatial navigation. So what do I mean by that? The classic tasks in decision-making have often been studied with really controlled and often limited movements, things like saccadic eye movements, nose pokes, or licks. The reason for this is to try to separate out potential movement confounds from cognitive processes like decision-making. So if we look at classical paradigms like the random dot motion task, where a monkey sees dots moving to the left or right and then makes a saccadic eye movement to report which direction the dots are moving, or cases in which rodents might lick a spout on the left or the right to report their decision, or cases in which rodents might do a nose poke and then make limited movements to go to the left or the right. These are all incredibly powerful paradigms, and there's a lot of power in separating out kind of these movement components from the decision-making components. But these don't really get to the question of how decisions are made during spatial navigation. In addition, a lot of studies of navigation have really been focused on neural mechanisms for things like position and heading. So they're really famous examples of head direction cells, place cells, and grid cells, which really give quite strong information about where an animal is in an environment and how it's heading. But ultimately, this has to be turned into kind of a plan of how to reach a goal location and identify a goal location. So I would argue that there's relatively few studies or that have really focused specifically on the context of decision making during spatial navigation. And so to really tackle this question, a few years back now, we developed this virtual reality system to study these types of navigation-based decisions. The idea is that a head-restrained mouse can run on top of a spherical treadmill and is surrounded by a large screen on which we can display visual virtual reality environments. So here's a movie where the mouse is running on this one of these mazes, and so it's just a linear maze here where it can run to one end of the maze and gets a reward and then has to turn around. Well, what we like about this is that we have complete control over the visual cues that matter to the mouse. We can use a lot of great movement measurements to understand how the mouse is moving and running during these types of behaviors. And furthermore, it opens us up to really using two-photon imaging, electrophysiology, optogenetics, kind of all the circuit analysis tools that are available in the mouse. And so we can extend this to doing decision-making tasks during virtual navigation, where we can have a mouse run down a T-shaped maze where it sees a visual cue at the beginning of the maze, either a white cue or a green cue. And based on the cue, it has to either run and turn to the left to receive a reward or run to the right to receive a reward. And so here's just a movie of the mouse performing this type of task. So you can't see the mouse in this case because it's blocked by a microscope, but you can see that it ran past the white cue, enters this delay, and then at the T intersection, it has to turn to the left to get a reward. There's a brief intertrial interval. We give the mouse another trial. Here is another white trial. This helps you get a sense for kind of the timing of these types of trials and types of behavior. And so once again, it runs to the end of the maze, turns to the left to get a reward. Another intertrial interval, and I'll show you one last trial where here the mouse sees the opposite cue, will run down the maze, and in this case has to turn to the right to get a reward. Okay, so there are two questions that I want to pose following from this for today's talk, and one is, what is the functional organization of cortex that's underlying these flexible navigation decisions? So if we think about all these individual areas one by one, we might start to think about cortex as being organized as kind of a modular or hierarchical organization, where different areas might be assigned, different functions, and then these areas might be pieced together and maybe potentially what we would think of as like serial processing to try to, transform a sensory cue into kind of a motor action. But interestingly, recent work has shown that there are actually very highly distributed representations. Here I'm just showing you one recent example from Carl Dijsselroth's lab, where they did a simple go, no-go task and did calcium imaging across a lot of different parts of cortex. And shown here on the different colors, you can see cells from different parts of cortex, and you can find cells in many different areas that seem to have similar activity patterns. So in the first part of today's talk, I want to talk about how can we maybe begin to reconcile or bring together some of these ideas of modular organization and these highly distributed representations, and just try to clarify what type of organization we actually see across cortex for these flexible navigation decisions. Then the second part of today's talk, I'll try to focus on the question of what are the causal relationships between cortical areas and navigation decisions. We don't just want to understand the representations of these types of variables, but we actually want to understand causal relationships. And as a start to this, in past work, we've inhibited posterior parietal cortex during this simple task that I showed you earlier, and we found that the mouse could perform this task at about 90% correct. But when we injected mucimol, this GABA-A receptor agonist, into posterior parietal cortex, behavioral performance went down to chance levels, indicating that PBC was necessary. And we can actually compare this to other tasks where, like, the mouse has to just run to a tall tower that's at the end of the maze. So it's a much simpler task, and in this case, PBC did not seem to be necessary. And so we want to build from this, and also build from really beautiful recent work from Lucas Pinto, Carlos Brody, and David Tank, where they've kind of extended this to evidence accumulation tasks and also many different cortical areas with optogenetics methods. And so we want to see if we can start to develop systematic ways across many different behavioral tasks in which we can start to uncover some of these causal relationships. So in the first part of today's talk, I want to talk about mapping the functional organization of posterior cortex and trying to develop a working model for this organization for flexible navigation decisions. This is the work of two really talented graduate students in the lab, C.E. Zhang and Salman Shte. And Salman is now a postdoc at Columbia, and C is still a grad student in the lab. So what C and Salman did was they developed this flexible decision-making task during virtual navigation. The idea is that the mouse runs down a maze. It sees either a black or a white cue. And then in what we will call rule A, the mouse can learn that the black cue means it has to turn up to the left in order to receive a reward. And if it sees the white cue, it has to run and turn to the right to receive a reward. But we play a trick on the mouse. So after about 100 trials within a session, we actually switch this relationship into what we would call rule B, where now the black cue means the mouse has to run and turn to the right to receive a reward, and the white cue means the mouse has to run and turn to the left. So we do this switch multiple times within every session. So the way this actually looks is it's shown as a Y-shaped maze where the mouse runs down the stem of the Y and then has to report its decision by going into one of the two sides of the Y maze. And then it enters a feedback period where it receives visual feedback and reward feedback about whether it got the trial correct. What we think is really interesting about this type of task is that as the mouse is running down this stem, the mouse has to combine its visual information about what cue it's seeing with the internal representation of the rule. We don't actually signal to the mouse which rule it's in. It has to learn that and memorize that through trial and error over some trials. and then use this combination of cue and rule to inform its upcoming action. And then at the end of the maze, in the intertrial interval, the mouse has to evaluate what cue it saw, what choice it made, and whether it got the trial correct in order to update the rule. And this is especially important around the times where we switch the rules. So this is what behavior looks like on an example session, where we start in rule A and the mouse can learn it's in rule A and start to do quite well, where it's doing about 100% correct. Then at the gray dashed line, without telling the mouse, we switch the rule to rule B. The mouse actually drops below chance levels, below 50%, because it's actually still doing rule A. And then over time, it learns that it's in rule B, and over tens of trials, it actually starts to perform nearly perfectly on rule B, and then we can switch the rule again, and then it can switch back to rule A. And this is what the behavior looks when we align all the trials together around the switch point, where you can see the mouse does really well up to the switch point, drops below chance, and then recovers over tens of trials. So one of the things that we really noticed from this type of task is that given these switches and these different rules, we actually identified quite a lot of behavioral variability. We've done a lot of work to actually model and understand the behavior of the mice during this task. I don't have time to go through all of that behavior modeling that we've looked at, but I want to emphasize one thing that we think is quite interesting. And that's the idea that we think we can start to estimate the time course of choice formation by looking at the running patterns of the mouse. So as the mouse is running down this Y-shaped maze, we can actually measure its running velocity by looking at the rotational velocities of the ball on which it's running, about 3 axes, the pitch, roll, and yaw axes, as it runs down the maze. And so if we look at individual trials here, where we look at just the roll velocity, which controls whether the mouse moves to the left or the right, we can plot individual trials in the thin lines in the center as a function of position within the maze. And so we can color code those by the blue where the mouse turned to the left and red where the mouse turned to the right. And so you can see lots of variability in kind of the running of the mouse on these left and right trials. If we look to the right panel, I've then set the means for the left and right trials in this thick blue and red lines, and then I'm showing 4 individual trials in which the mouse turned to the left. And we can just kind of illustrate this variability, where on trial number 1, the mouse actually turned to the left very early on and kind of ran to the left throughout, whereas trial number 4, the mouse actually turned to the right, and then only very late actually corrected and turned to the left in order to complete the trial. So we actually saw quite a bit of variability, and we wanted to see whether this variability could be related to actually the upcoming choice of the mouse. So we tried to predict the mouse's final reported choice on a moment-by-moment basis based on these running trajectories. So the way we did this, there could be multiple ways to do this, but the way we chose to do this was with a small recurrent neural network, here using a long short-term memory network in LSTM, in which we fed in the full trajectory of the mouse's running up into a given point in the trial. So we did this at every single point within the trial, feeding in the full trajectory of the running up into that point. And then based on this, tried to predict whether the mouse would ultimately make a left or a right turn at the end of the maze. So we're basically at every time point based on the running trajectory trying to predict whether the mouse will eventually choose left or right at the end of the maze. And so we end up with a variable that we can call dynamic choice. We think of this as the probability, or quantify this as the probability that the mouse will turn to the left or the right. So at every position in the maze on every single trial, we can actually estimate this probability. And so on left trials shown in blue, you can see that this kind of evolves to the left relatively early on. And then this probability of left actually goes very low on the right turn trials. And we can once again look at these individual trials and kind of recover this type of variability that we see. But we think what's quite exciting about this is that this dynamic choice gives us kind of a behavior-based estimate of choice formation. Now, I don't have time to show you that we've tried to verify that this actually is a sensible type of variable. I'll just explain this briefly, where we've also done this completely separate type of behavior modeling where we try to estimate the mouse's strategy on a given trial. For example, it could make rule-based decisions where it's using the cue to inform its choice. It might actually be randomly guessing, or it might have a bias of turning to the left, for example, instead of turning to the right. And so we've developed behavior modeling where we try on every trial to estimate the strategy of the mouse. And what we find is that on the sensible relationship with this dynamic choice, where the mouse tends to be following the rule, we see that the dynamic choice tends to ramp up relatively quickly. when a mouse seems to be making random guesses, we actually see that this kind of hovers around like 0.5 and then comes up very sharply very late in the maze. And then if the mouse is very biased, for example, we might see that that this probability of going one way is actually very high at the very beginning, as if the mouse has already made up its mind even before having time to interpret the cue that's present. So we think that this is an interesting variable. And importantly, I want to note that even though it's based on the movements of the mouse, it's using kind of the integrated trajectory of the mouse, and thus can actually be dissociable in some ways from the instantaneous running patterns of the mouse, which is a point we'll come back to when we look at the neural encoding.
Chris, we have a question.
Yeah.
I think maybe now's a good time for it. So this is Tom Crew asking, I'm going to ask for him. The question is about metalearning. You have blocks of trials where you're switching implicitly the rule. And Tom is asking whether you can see any evidence for any metalearning, i.e. switching faster or changes in these trajectories with experience.
That's a great question. Yeah, we've looked at that some. We were thinking that maybe the mouse would start to switch faster as it as it got further along in training and so on. We didn't see strong evidence for that actually, especially after the mouse gets to be kind of expert at doing some aspects of the task. We didn't really see like a major increase with the speed with which it can switch. So we don't know exactly why that is. There's a lot that goes into these switching cases where errors could arise because the mouse forgets what the sensory cue is and so on. So it's not always performing 100% correct and so on. But I would say didn't see major evidence for that type of meta learning. But it's something that we could definitely investigate a little bit further, I think.
Thanks.
Okay, so we then turned to wanting to understand some of the neural activity that's present, and so we thought we could do calcium imaging.
I'm sorry, Chris. We have a follow-up question. I feel like this is a transition worth dwelling on, if you don't mind. So the follow-up question relates to how the mouse is making the decision. I'll read it to you. Rather than maintaining a representation of the rule, could the mouse simply be updating and maintaining action values at each state?
Yeah.
For example, well, I don't know if I need to translate that for you further.
Yeah. I think
the question. Well, I'll let you continue.
I don't know if this will fully answer it, but I think yeah, there has to be whether how the rule is represented. I think is a good question, right? And it doesn't have to be an explicit rule type of representation, right? There can be, but the cue has to be associated with some action in some way. And what we like is that we've totally balanced this in a way that every action has the same value to it, right? We're not changing reward probabilities or things of that sort. So left and right actions are always, have equal likelihood of reward across all the blocks. But I think there's lots of different ways in which this type of rule could be represented, and that's something where we don't have a good answer for yet, but something we're thinking about more from the behavioral side and also from the side of looking at how things are encoded in neural activity.
Yeah, I mean, to me, it seems, if I could just sort of engage a little further, to my mind, it seems that since the rules are blocked, you're obviously masking the feedback. You're masking the rule with the feedback, because the feedback is consistent from trial to trial if it goes to the right. This action value idea. You could randomize the rules trial by trial. That would presumably make for quite a difficult task for the mice to perform, because the rule would be, explicitly then need to be cued and changing each time.
Yeah. We chose not to explicitly cue the mouse to the rule. So we couldn't really do it switch trial by trial. Yeah.
Right. So then the alternative, I guess, means that you would need to potentially do extinction tasks, where you remove correctness in the feedback and see how perhaps that alters future behavior. Do you think there's a way you could get a handle on this behaviorally in your system?
Yeah, I mean, I guess we could try some of those extinction experiments. I'd have to think exactly what those would be testing, but Yeah, I think it's a good topic for further discussion. We haven't done anything in that direction yet, so I think there could be opportunity to think about things along those lines. But I don't think we have much to say on it at the moment, given this is the only variant we've tried thus far.
Okay, great. And we have a methodological question before I leave you alone for a little bit again. The question is asking about in the dynamic choice model, what are the labels for the time points before the decision? In the sense that maybe the animal hasn't chosen yet, do you define the decision, the labels based on the future decision, or is there another way you choose the labels before the decision?
We're trying, yeah, we're trying to based on the running trajectory up into that point in the maze, we're trying to predict the final choice that the mouse reports at the end of the maze. So what we call the reported choice, whether the mouse eventually chooses left or right. So what we're trying to get at is sometimes you might not be able to predict very well which direction the mouse is going to turn, in which case we would say that perhaps the mouse has not yet committed to a choice. Whereas other times we could predict very well which way the mouse is going to turn, in which case we would say that perhaps the mouse has committed to a choice already. So we're trying to use that type of like how well we can predict based on the running trajectory, which way the mouse will ultimately report as its choice, an idea of kind of the dynamics of this choice formation.
Fantastic, thanks a lot.
Okay. So yeah, so we turned to calcium imaging, and we thought about multiple ways we could go about trying to understand neural activity here. And so one way we thought was, we could do what's maybe most standard and what we've done in the past, which is go to some areas of interest and try to record activity there. So go to posterior parietal cortex, go to visual cortex, go to retrosplenial cortex, and record cells and kind of lump them all into that into those area boundaries. But we thought we would take a different approach here, where we would sample neural activity without respect to area boundaries. And one of the reasons we did this is because actually some of the area boundaries in mouse posterior cortex are not so well defined. I think you can define very well the boundaries between V1 and other areas, and maybe retrosplenial and other areas, but a lot of the what we would call posterior parietal cortex areas don't necessarily have strong anatomical or functional boundaries to them. And thus we thought perhaps the better thing to do would be to sample neural activity without respect to these boundaries. So what we did is, on a given day, we took a single field of view where we did volumetric 2-photon calcium imaging. We then took an overview image that looked at the blood vessels. We could then zoom out and look at our total cranial window and line up the blood vessels. And then we could identify the location of every cell within this field of view. So just to orient you, this is anteriors towards the top, posteriors towards the bottom, medials to the right, and laterals to the left. And so like the bottom left of this yellow box would be V1, and the most medial part would be retrosplenial cortex. And then what we could do is then just kind of on different days move around our imaging window so that we could kind of tile across this entire space and just collect imaging data across this entire space. And then we could then do retinotopic mapping, where we could identify different retinotopically defined cortical areas, such as V1 in the bottom, and like that other red blob that comes off to the top right as like area AM, these areas that people have defined as like secondary visual areas. And then we could align these retinotopic maps to the Allen Institute Common Coordinate Framework. So what we can do is then basically align all of our imaging field of view to this fields of view to this common coordinate framework, and thus identify put every single cell that we imaged into the same common coordinate frame. And so of course, if we wanted to, we could then still divide cells up into different areas. We could divide them into retrosplenial cortex V1, this anterior area A, which we can think of as like the anterior part of PPC, and then these other areas like AM, MM, and PM. And so Bijan and I were talking earlier about how one might define where PPC is and where all these different areas, what these different areas are. And I think that's a really great question, and one we're still trying to get at. But I would say that kind of the functional boundaries within these areas are are not always completely well defined and could depend on different tasks. And so I think it's still an ongoing question. It might always be kind of an ongoing question where there's not really sharp anatomical boundaries between these areas. So the data set that we got, this is all done with viral expression of GCAMP6S. On every single session, we collected hundreds to thousands of layer 2-3 cells simultaneously. The overall data set includes about 300,000 neurons that were imaged over about 300 sessions in 8 mice. I really want to emphasize that this 300,000 neurons is what we think is kind of an unprecedented scale for decision-making tasks of this sort, where you have this across many areas and many different neurons. And we think that is really quite powerful and opens up the doors to a lot of different analyses, of which I'll show you some that we focused on. So of course we can go and start to look at individual cells within these areas and see what their activity patterns look like. I'll show you 3 such examples of these cells. So here I'm showing you on each row on the left here is a single trial. I've grouped those by trials in which the mouse held a black cue and turned left, black cue and turned right, and so on. And so this neuron 1, you can see, is really active at kind of the beginning part of the maze, and only when the mouse saw the black cue. So we might think of this as kind of a cue selective neuron. We can see other neurons like this neuron 2 that was active only when the mouse saw the white cue and turned to the left. So we can think of this as kind of maybe a cue and a choice conjunction. It also has some rule information to it because it's only active on one rule and not the other. And then we can see a third neuron like this that was active in the feedback period, where its activity seemed to be active kind of across all the different trial types in the feedback period, and it might be a little bit harder to understand exactly what this cell was related to at first pass. But of course, looking at these single neurons is great, but we needed a more systematic way in which we could begin to analyze the population. So we turned to a generalized linear model, a GLM, to try to quantify the encoding profile of single neurons for multiple task and behavioral variables. So the idea is that for every neuron, we have its activity profile across a session. And then we can use as predictors of that activity all the different task variables that we measure or control, like the cue the mouse saw, what choice the mouse reported, the outcome of the trial, whether it was the previous trial information. We can include some of these strategy variables, like I mentioned, things like whether the mouse was biased or randomly guessing or following a rule. We can also include this dynamic choice variable. And moreover, we can include instantaneous movement variables, the instantaneous velocity and acceleration of the mouse, and actually develop through this B-spline basis functions actually kind of allow for complex types of movements to also be represented. And so we did this type of model for every single cell in our data set. And so just for those 3 neurons that I showed you before, here we can look at the quality of the model fit for these different neurons. In black, I'm showing you the kind of left out data, so data that was not used in the fitting of the model. And in red, I'm showing you the prediction. And we can quantify that for this metric deviance explained. This is conceptually related to something like the variance explained, but this is a metric for how well the model's doing. And all these values are reported kind of on a single frame by frame type of prediction. Then what we can do is we can then start to remove in various ways groups of these different variables from the model and see how that affects the prediction quality of the model. And so then we can, by removing some of these variables, we can then start to, or zeroing some of these variables, we can start to understand how much they might contribute to a neuron's activity. So when we do this, we find that this neuron 1 that I showed you at the beginning had a lot of contribution from cue, as you might expect. It looked like one of these black cue selective cells. Neuron 2 seems to have both cue and choice, which which we might expect because it was active on only these white trials with left turns. And then neuron 3 that was active in this intertrial interval actually seems to be most related to the pitch or the running velocity of the mouse. So we can start to do this then across many different neurons and across space. So we know the position of every neuron and we have its encoding profile. And so we can start to look at specific variables of interest. So we can start to look at the instantaneous movements of the mouse. So what I'm plotting here is a plot where each dot is a single neuron. We're color coding that by how much of its activity can be explained by the instantaneous movements of the mouse. And just to orient you again, kind of at the bottom left is V1, To the right is retrosplenial cortex. And then at the very top would be like the anterior part of PPC, this area we call area A. And so you can see that that movement is most highly represented in this anterior part of PPC, as you see the most yellow up there. And you can see that actually, if we smooth this plot just to show the general trends, you can see this kind of anterior to posterior gradient of having the highest movement in the anterior part of this posterior cortex. But what you also notice is that movement is kind of represented everywhere to varying degrees. So this is consistent with much work that's emerged in the field from multiple groups that has shown that movement can be present in many areas. But interestingly, we found that it was not uniform. We found this gradient with the strongest movement encoding in the anterior parts of PPC. So we can, of course, do this for all the variables that we looked at. I'm not going to show you the maps for every single variable. I'll just point to two more. We can also look at the identity of the visual cue. And as you might expect, this is also that is represented strongest in V1 and kind of fades off going into secondary visual areas. And so this is now looks to be kind of the opposite type of gradient of movement, where it's highest in the posterior parts and fades off going more in the anterior direction. And then finally, I'll show you just the map for dynamic choice. This choice signal that I mentioned earlier, if you look at the scatter on the left, you can see that it's relatively highly distributed. But when we look at a smooth map, you can kind of see that the highest representations are actually in retrosplenial cortex. This was pretty interesting to us because retrosplenial cortex is not an area that's typically thought of in terms of decision making per se. So it was interesting that that's where we saw kind of the highest dynamic choice signal. Now I'll note that the magnitude of the signal is a little bit lower than what we saw for other variables like the Q. We think this makes sense because the Q is something we as an experimenter are controlling and can make very salient, whereas dynamic choice is a more cognitive variable that we're estimating with our behavior modeling, and thus might be a little bit harder to get the right estimate and so on. But we found it interesting that we saw kind of this medial to lateral type of gradient for encoding of dynamic choice with the strongest in retrosplenial cortex. So when we put this together, we just looking at these three features, we can see three prominent spatial gradients with cue, movement, and choice. And interestingly, the choice and the movement type gradients seem to be peaked in different locations. Once again, pointing out that dynamic choice can perhaps be separated from this type of movement. So we wanted to understand whether these were like real and important types of gradients that were present. So we thought of several ways in which we could begin to test that. So first, we thought that if these gradients are real, then that means that cells in different parts of posterior cortex should have different encoding profiles and thus encode different features. So we thought if this is the case, then we should be able to take the encoding profile of the neuron and actually be able to predict where in anatomical space it resides. So what we did was we took these encoding profiles for each neuron, like I'm showing you on the left here, where how much of its activity could be explained by each of the different groups of variables. Then at a set of discrete locations within the posterior cortex, we then developed these decoders to ask what's the likelihood that a cell with that type of encoding profile would actually be present at that type of location. And we did this across all the different spots. So what we did was we took, let's say, a V1 neuron and asked what's the probability that that V1 neuron would reside at all these different parts of posterior cortex. What we found is that V1 neurons we could actually predict to be most likely to be present in V1. If we looked at area A cells, we found that those cells were most highly predicted to be in area A. And retrosplenial cortex, those cells tended to be most highly predicted to be in retrosplenial cortex. This is basically saying that there are some distinguishing features of V1 cell activity, RC cell activity, and area A cell activity. Interestingly, when we looked at the areas that were more in the middle, like these areas AM, PM, and MM, it was much harder to actually predict that cell's location. And this is probably likely because this is where all the gradients came together, and thus there was a lot of these different features that were present. And so it was actually much harder to decode that cell's location based on its encoding profile. So we think this shows that these encoding gradients are actually real things that that are significant features of the data. But I'm basing them on three features that we've kind of hand chosen. So can we actually identify these gradients in a more unbiased way? So what we did was we turned...
Chris, I'm going to choose to interrupt you at this moment.
Yep.
I feel like you're probably going to answer these questions in various ways moving forward, so I thought I'd ask the question that you handle how you want to respond. The first question is essentially asking about the decoding labels you've defined and whether they're dependent upon each other. So you have visual derived labels, the Q ones, you have motor derived labels, the choice ones, the movement ones that lead to the choice itself. And the question is basically pointing out, could you have optic flow, which is a visual signal that's generated by movement, soft motion, of course. And do you think, well, is it possible in your experiment to start with? Do you think you see optic flow signals is the question?
Yeah, there's exactly those could be perfectly related to each other, but we have an interesting point where we have in the intratrial interval, for example, the mouse is still running, but the screen is dark, so there's no optic flow, but the mouse is still running. So that gives us a point where we can actually very neatly separate out movement and optic flow. So if a cell is tuned to the movement of the mouse, then it should be doing that both in the intratrial interval and in the maze, right? So we think we have a good way to separate those variables out through, for example, that different part of the maze. Yeah.
Okay, good. Thank you. That sounds like a great control to me. The next couple of questions are essentially asking about the figure we're watching in front of us and highlighting this issue that you've already referred to, which is that the fractional explained deviance of the dynamic choice variables is much smaller by, I don't know, a factor of 10 or so up to than the other signals. So one way of asking this question is, how would we see the right-hand-most result if we were to plot it on the same color scale as the left-hand, one of the other two?
Yeah, I think well, we could do that, and I think you can imagine, but I think, yeah. So I think there's reasons why we would not actually expect it to be as big as these other features, but it's also true it is lower. I'll just say to kind of calibrate you a little bit on what do these things mean. So if we took, let's say, 100 nearby cells and tried to decode the choice or dynamic choice from the activity of those cells in retrosplenial cortex, we can do that quite well. So these are, it's not to say that this is like a totally meaningless magnitude of a signal. Actually, from this type of signal from nearby cells, you can actually decode quite well. I'll also say that some of this is the average across all the cells that we're looking at in this analysis, and so there can be nearby cells that some encode the choice strongly and others don't, and then you average those together and you get kind of a weaker looking signal. And it tends to be that there are some cells that just really strongly encode the dynamic choice and others that don't, and the density of cells that really strongly encode the dynamic choice tend to be a little bit lower than those for these other parameters. So that also contributes to this value. So I think it's not exactly trivial to interpret the meaning of the magnitude of this, but we interpret it as substantial in the way that, like, at least you can decode these variables quite well from a population of neurons.
Okay. So this area of population decoding and coding, I think you're probably going to talk about more as you move forward. So I'll hold off on any other questions in that direction. I think one question I have about this that came out of your sort of this, maybe asking this most recent question again in a different way. The Q cells, is there fractional explained deviance in the retrosplenial cortex? The Q result for the retrosplenial cells, I don't know what the value is. It's obviously much smaller than it is in the visual cortex. Is it smaller than what you see for the choice variable in those neurons? I don't know that you've explicitly. I mean, I guess it.
Yeah. So yeah, it's hard to see that here. It turns out that it's similar scales actually. That retrosplenial cells would have similar scales of kind of Q and choice encoding on average.
Okay, and so this is kind of point. This question is echoing, but I think I should just acknowledge it. This is pointing to this issue of how the choice variables may be in some ways correlated in a manner that you can control, perhaps, but hasn't been completely controlled in all cases, with the other motor and sensory variables. And maybe that's. Do you think that that's consistent with the explanation of the Q-select of these responses in the retrosplenial cells to visual and motor signals? Is that where your thinking is?
Yeah, I'll say Q and choice are perfectly decorrelated by our task design with the rule switches, right? So that's one thing that was a big motivation for actually this task. So we don't think that should really explain these types of things, but there are correlations between variables and so on. So I think that could contribute, and the degree to which that's contributing, I think, is hard to fully appreciate. And so yeah, so we don't get completely hung up on really comparing all the magnitudes of these things in great detail, but have focused more on kind of the relative encoding of these across the different parts of cortex.
All right, thanks a lot for those answers. Let's let you two continue for now.
Okay. Yeah, so I was saying that we've looked at these three variables, right? But and looked at their gradients, but are these gradients actually really dominant features of the encoding profiles across posterior cortex? So what we did is we did an unsupervised factorization here. We actually took the encoding profiles, actually building from these decoders that I just mentioned. I'm happy to go into the details later, but essentially we're taking the encoding profiles of the different neurons and then factorizing them, and then looking at kind of how these factors where they have the strongest in anatomical space, where they have the strongest weights. And so we can find three factors that actually neatly line up with what we think of as the visual, the choice, and the movement type of gradients that we observed. And we actually find that three factors explains a lot of the activity across space quite well. So we think that actually by doing an unsupervised factorization, we can actually uncover these three types of gradients as well, kind of lending credibility in our mind to the idea that these could be prominent features of the activity. So we think this is interesting because this starts to show kind of prominent organization to the posterior cortex, but I don't want to give you the impression that everything is kind of organized into these like more compartmentalized type of activity modules. It's actually quite the opposite. Representations are very highly distributed. So if we go back to these scatter maps for the cue, movement, and dynamic choice encoding, you can see that these are actually, very highly distributed for all variables, and also cells that are nearby one another can encode things in very different ways. And what distributed representations mean is each variable is represented in lots of different areas, but also that each area is going to then have multiple variables that are represented. So we thought, what importance could this have, and how could we start to relate this back to some specializations? And one thing that we noticed and thought was really important is that rather than just looking at these individual variables, we could start to look at conjunctions of different variables. So looking at the representations in single cells and finding that they're often conjunctive for multiple task and behavioral features. So the idea here is I'm plotting a plot of the, how movement related cells are versus how they're related to task variables, things like cue and choice. And so in this density, you can see where cells in these different areas fall. So in V1, they tend to fall more in the upper left, which would be less movement and more related to the task variables, probably cue. Area A is more on the bottom right, indicating that it's more related to movement and less to some of the task variables. But in retrosplenial cortex, for example, we find a large density in between, indicating a conjunction of these different variables. I can show you something very similar where we look at this for cue and dynamic choice, and now we actually find that in all areas, maybe most in retrosplenial, but definitely in all areas, some degree of cells that are kind of falling along more of the diagonal, which would be kind of a conjunction of the cue and the choice that are present. And so I don't have time to go through all the different possibilities here, but I'm just trying to illustrate that we actually find that representations in single cells can actually be quite conjunctive, and that this conjunctiveness can actually vary between the different areas. And so we just wanted to kind of formalize all of this in a little bit more quantitative detail. So we came up with a way to quantitatively compare the encoding across cortical space. And so the idea here is we take the encoding profiles of different neurons. I can go into methods later offline for those who are interested, but we essentially come up with a linear embedding where we embed every single neuron in this space. And so each dot here in these maps is a single neuron. And so neurons that are close to each other in this space are expected to have similar encoding profiles and predicted to be similar to each other in cortical location. And then we can overlay on that the scores for these different factors from that we got from this unsupervised non-negative matrix factorization. And so we can find one part of this kind of density that looks more like factor 1, which was more like the visual factor, one that looks more like what we call the factor 2, the choice factor, and one that looks more like factor 3, which is more like the movement factor. But what's really interesting is then we can start to overlay on this the contours for all the different cells from all these different areas. And what we can find is that in brown for area A, you can see that its density, its contour sticks out most towards this factor 3, whereas retrosplenial in purple sticks out most towards this factor 2, and V1 sticks out most towards this factor 1. But what's striking is that even though these kind of stick out and have these maybe specializations, most of the density for all these different areas is more in the center. Thus, there's really extensive overlap in the encoding across these areas, which can lead to these highly distributed representations or emerge from these highly distributed representations. And due to this overlap, at the center, kind of between these three gradients in this encoding space, we might expect there to actually be extensive conjunctive coding. So these are things that we're actually exploring more at the moment in more detail to understand in more depth. But I think just to summarize, we think that there are three key features that we need to take in mind for this functional organization of posterior cortex. First, we have this functional specialization where we see these gradients of encoding that go in different directions. We see these highly distributed representations. And furthermore, we see the integration or conjunctive coding that's present within single cells. And so we think that these are kind of the ingredients that need to be put together to understand kind of some of the function of the posterior cortical areas. And so we're moving with this, and we're excited about this is that we think that this starts to allow us to build a working model for flexible navigation decisions based on an idea that we're thinking of as something like integrative specialization. The idea is that, in very cartoon form, we can have maybe three different areas that have distributed encoding and all encode various aspects of visual choice and movement information to different degrees. But we know that kind of the ratios with which these types of information might be combined could be different between these different areas, indicating that maybe these cells, these areas integrate information kind of different ratios for potentially different functions. And this can maybe lead to ideas about predictions for the computations that are present within these different areas or the functions of the different areas. For example, we might think of V1 as being important for visual computations, as you might expect. We might hypothesize that retrosplenial is actually important for forming navigation decisions. And this area A may be more for movement biasing. But we don't think of this as kind of this feed-forward serial type of processing. Rather, we think there has to be really important recurrent interactions where once a navigation decision is made, it has to update the area that biases the movement. But then once that movement is biased, it can also come back to help form and update navigation decisions and so on. And so we're excited because we think we now have a data set where we've imaged enough areas, or a lot of areas at least, in the same context with the same behavioral tasks, same animals, same analysis methods, and so on, that we can start to put these areas together. And so where we're going with this is we're starting to build large dynamical systems models, including inspired by some of the work from Xiaojing Wang's lab and others, to really start to put these pieces together, inspired by the data that we've collected, so that we can hopefully come to maybe mechanistic predictions about how these areas operate as a dynamical system and begin to actually carry out some experiments to to test those types of predictions. And so with that, that's the first part of today's talk. There were quite a few questions, so things have gone fairly long. I will ask Bijan his impression on whether I should continue with the second part of the talk, which is relatively short, or whether we should kind of call it at this point.
Please continue. We're very interested to hear the second part of your talk. You have, well, people will leave when they leave, but we have another 15 minutes, I would say, if you need it.
Okay, great. Cool. Okay, so yeah, so the first part of the talk was talking about looking mostly at the encoding and the representations, but that's only part of the story, right? We also need to identify some of the causal relationships between cortical areas and decision tasks. And so the idea is that we basically, we being Charlotte Arlt, a very talented postdoc in the lab, and Roberto Barroso, a former research tech in the lab, we really wanted to see if we could come up with a systematic way to try to understand how these cortical areas could be related to navigation decision tasks in a causal way. And we came to kind of what we think of as a surprising and interesting finding, that there can actually be quite flexible neural implementations that depend on prior experience and cognitive context. So I'll unpack what I mean by that. So we were inspired by asking the question, are cortical areas necessary for these tasks? And if so, what specific aspects of these tasks? We want to say, for example, can we assign a specific label for what aspects of the task retrosplenial cortex might be necessary for, as opposed to area A of PPC and so on. So we thought about how we could do this. We wanted to take the approach where we tried to inactivate these areas during different behavioral tasks, and we thought about ways in which we could vary those tasks. We thought we could perhaps vary the sensory and the motor aspects of the tasks, but we instead chose to kind of try to have mice perform what we might think of as a similar or identical navigation decision that involves the same sensory cues and the same locomotor actions, but just put this into distinct cognitive contexts and see how that affects the involvement of these cortical areas. So as a platform for this, we developed a really simple navigation decision that we use as a substrate for testing these involvement of cortical areas. This is once again a Y-shaped maze. Here the mouse saw one of two visual cues, either horizontal bars or vertical bars. These were present throughout the maze, and the mouse just had to make the simple choice of going to the left if it saw horizontal and to the right if it saw vertical. So there's no delay periods, there's no switching at this point and so on. It's just kind of a simple type of task. So then we turned to optogenetic silencing, following work that was done in the Svoboda lab and other labs, where through the skull in a mouse that has channel rhodopsin expressed in inhibitory interneurons, we could shine blue light from a laser at different spots. So focusing on PPC, retrosplenial cortex, or anterior cingulate cortex as kind of prime association areas in cortex in the mouse. And then we also included a spot in somatosensory cortex and a control spot that was actually outside of the brain. And we inhibited these areas basically throughout the full duration of the trial. So when we inhibited these areas on this simple task, we actually found that there was really only a moderate effect on behavioral performance. So the mouse could perform this task at about 95% correct or so. And then when we inhibited areas like PPC, for example, the mouse could still do the task at about 90% correct. There was a statistically significant decrease in performance, but it was relatively moderate. So we kind of concluded that the association areas were not really essential for solving these types of simple tasks. So we thought, what if we changed the cognitive context in which this navigation decision was made? So we returned to our rule switching task, this what we call the flexible context here, where now just using different cues, the horizontal and vertical, but the same idea as from the first part of today's talk, where there were two rules in which the cues and their choice directions were switched between those rules. But what's really important here is that in this simple context where there were no rule switches, and in rule A of the flexible context, we used exactly the same VR software code, meaning the same sensory cues and the same choices that were required for the mouse, but they were just embedded in these different contexts where the mouse either had to perform the rule switches or did not. So here I'll just show you on the left an example movie of the mouse solving one of these trials in a simple context. And then on the right, I'll show you a different mouse solving the same trial type, but now in a flexible context. And the whole point of this is that these things look, identical. You can't really tell them apart from the perspective of the cues and the choices the mouse has to make. But now when we inhibited areas in rule A of this task, we've now saw dramatic effects on the behavioral performance. We did this not around the switch periods, but around the periods where the mouse was having strong performance well after it had already switched. And so we found when we inhibited places like RSC, PPC, and anterior cingulate, behavioral performance dropped to really around chance levels, meaning the mouse could no longer perform these types of trials. If we compared that to the simple context, we saw much bigger effects. Here I'm plotting the delta fraction correct relative to control. So how much did behavioral performance decrease relative to control when these areas were inhibited? And you can see that there was a much bigger decrease with the flexible context than with the simple context. So we thought, okay, this we think is interesting, but is there something specific and special about this rule switching and flexible context? So we turned to a different type of task, taking advantage of something that's very common in systems neuroscience, and that's adding a delay period between the sensory cue and the ultimate choice. So here was the identical sensory cues again, the horizontal and vertical bars, identical choices. The mouse has to go to the left or the right. And we just inserted a delay period that was like a gray part of the maze between these two. Now, when we did this, we once again saw that when we inhibited these areas like RSC and PPC, the mouse could no longer do this task anymore, indicating that these areas were really critical for the mouse to perform these types of trials. And once again, this was much bigger than when we looked at this in the simple context. So we think this is starting to be interesting, where with the same cues, the same locomotor actions to report the choice, but just changing the cognitive context had a much bigger effect on which cortical areas were involved. So we've seen this now in two cases with this rule switching and with the delay, and we thought, is there some commonality between these? And the only commonality we could actually come up with was that maybe there's potentially short-term memory in the delay case and short-term memory of the rule in the flexible context. So we then tested a more complex case, which we call the diverse context, that did not have any what we could perceive as a short-term memory component. So this just had two more trial types where the mouse could see a black cue and go left, or a white cue and go to the right. And so all these trial types, there were now 4, but they were all kind of independent of one another and just randomly interleaved with one another. And now when we inhibited these areas, we once again saw a major decrease in behavioral performance when we inhibited retrosplenial cortex and PPC, and maybe a little bit less so anterior cingulate cortex. But once again, these were substantially bigger than just looking at the simple context. So we think this is exciting, and this builds off of previous work from Lucas Pinto, showing that if we change the cognitive complexity of the task, but here importantly keeping the sensory cues and the locomotor choices the same, just by changing the cognitive context, we can greatly vary which cortical areas are involved. But we didn't stop there. We did one more experiment, and this is the last final experiment I'll tell you about, where we tested what about prior experience and whether that can affect navigation decisions. There's beautiful previous work from Greg DeAngelis's lab that's shown that with higher experience, that the necessity of cortical areas actually decreases. This is an area MT in their case. In our case, we found that with prior experiences actually increased, the opposite, actually increased the involvement of cortical necessity. So what did we do here? We trained some mice just on the simple context. This is the context that doesn't require, for example, PPC to perform the task. Then we trained other mice on the flexible context, which does require PPC. And then we transitioned those mice to the simple context and had them just perform the simple context for weeks. So they have prior experience on the flexible context, but now for every day for weeks, they're just performing the simple context. So they know that basically no rule switches are happening anymore because they're doing this for weeks. And now when we inhibit PPC here in an example mouse, looking at the change in performance relative to control trials, we actually see that even after 2 weeks in this simple context, we see that PPC is still required for the mouse to perform the task. This is in striking contrast to the case with mice that just had the simple experience, where we now see dramatic differences. The mice, and these are different mice, but they're performing the identical task and they just have different prior experience. So we therefore think prior experience can greatly affect cortical involvement in navigation decisions. This wasn't the case on all mice, I should say. In some cases, the necessity of PPC kind of faded away over some time. But if we looked at this now across a population of mice, we could see, we've done this in 5 mice to date, and 3 of them showed this really strong effect of kind of maintaining this involvement of PPC even after they've been transitioned to the simple context. Therefore, we think that prior experience can greatly affect cortical involvement in these navigation decisions. So what we think is really, really interesting and exciting about this is that, we set out to, and challenging, I should say, is we set out to really study what's the specific role of a given cortical area in these types of tasks. And what we found is that that's maybe going to be a challenging question to address, because it looks like past experience and various forms of cognitive context can really alter cortical involvement in these identical navigation decisions, identical meaning same sensory cues and same locomotor actions. We think, therefore, this idea of converting a sensory cue into a locomotor action can actually have very flexible neural implementations depending on this past experience and context. As I said, it might be then challenging to really, with this type of method, isolate the specific computation that a given area is performing, and rather this method might really need to be refined in other ways going forward to really get towards those types of questions. And we think that just from a practical perspective, this really starts to illustrate that it's critical to report, control, and really test the effects of context and past experience on neural activity. This isn't something that's always done in all studies, and we think can have really dramatic effects on which areas are actually involved. And Charlotte has actually gone forward and actually looked at neural activity and also affects the neural activity patterns within areas. So I'll just wrap up quickly by giving you 2 hints of directions that we're going. So how can we tie all these areas together in more meaningful detail? Charlotte and Sophia in the lab are now doing mesoscope imaging, where we still have cellular resolution, but are looking at all these cortical areas simultaneously so we can understand how their activity is coordinated on a moment-by-moment basis. We're hoping to take these types of data and help to build into build them into large dynamical systems models that might give us mechanistic predictions. And then on the causality side, we're moving forward with more refined manipulations with single neuron and small groups of neuron photostimulation to try to understand if we can test causal effects on behavior, but also understand how local cells might influence their neighbors. So we're doing this with 2-photon optogenetics. This is work from Dan, Anna, and was set up initially by Salman in the lab, where we can go to single cells here, like target the blue cell and with 2-photon light and redshifted opsin, stimulate that cell, then go to the red cell and stimulate that cell, and then go to the green cell and stimulate that. And we can do that for single cells, and we've scaled up where we can now do this for tens of cells. And so I'm happy to talk to people offline about directions where we're taking that. So with that, I'll just conclude and thank all the people in the lab. This is a really talented group of people that I have tremendous pleasure and feel of working with, and I feel really lucky to have this group. They're a really dynamic, interactive group, and are doing amazing things that I'm quite excited to be able to work with them on. Especially want to thank Si Zhang and Salman Shte, who did the work for the first part of today's talk, and Charlotte Arlt and Roberto Barroso, who did the work for the second part of today's talk. And I'm happy to take any questions that remain, and I apologize for going a little bit over.
Well, thanks a lot. You're very patient with our questions, so I think that we should take the credit for the duration of the talk. So there's a couple more questions. One that I want to start with is clarification. The question is the nature of how the inhibition experiment was done. Were the inhibition trials interleaved with the uninhibited trials, or were they done in blocks?
They were interleaved, all interleaved, so all randomized with each other. Yeah.
Okay, so that leads to the follow-up then. Did you see changes in performance that dipped and recovered as you did the experiment?
Yeah, we just on those individual trials, we didn't see effects across trials, and we tried to be aware that we had a higher proportion of control trials so that we didn't really disrupt the mouse's behavior that much. So we, part of the challenge with those experiments is you can't get that many good inhibition trials per session because you don't want to really just strongly disrupt the mouse's performance. So we kept the number of true inhibition trials down to relatively low. How
low did you go? Do you remember?
I don't remember offhand. I think it was maybe in a scale of like 10 to 20% at the very most.
Thanks. So there's a couple other questions that are on the docket. One I feel you sort of answered, but I'm going to ask it again to make sure, because I feel like it's not exactly how it was posed. The question is about the first part of the talk, and it's asking how, well, the question is it the case that the activity of PPC neurons can be predicted by a distinct set of features, or is the activity of most of the cells not explained by most of the features? It's like your conjunction analysis, but posed in a different way.
Yeah, I think, there's definitely, I think we need to dig into that more, right? In terms of like what we can definitely predict to some degree where those cells will be located. So they do have some specific encoding profile that might be characteristic of those cells, and movement is one thing that comes out, but there's, digging into that a little bit more, I think is warranted to figure out what those patterns would be, and that could include and then seeing if like conjunctions contribute to that and so on. I think there's more to do there, but it's definitely the case that that we could, based on the encoding, predict some to some degree at least, the location of the cell.
And then the last question that we've got that remains to be asked is broader question, and it asks about the sources of the information, the possibility that other brain regions are, not in the posterior cortex, therefore, are providing inputs that actually carry the code or carry the signal that's being encoded. What thoughts do you have about that?
Yeah, that's a great question. We from the first part of the talk, right, it's just a purely looking at the representation and the encoding, and we have no idea where that information is coming from, right? It could be inherited from other areas. It could be generated within those areas. We don't know, and I think that's where zooming out to other areas can help. But also I think that's where some of these manipulations will be really powerful, if you can manipulate one area and look at what's going on in other areas and things like that, or through some of the modeling, maybe there's ways to look at Konica Rajan has these interesting models of looking at current flow and these large recurrent neural networks, and maybe those would lead to some predictions as well about where some of this information might be generated. But I think these are hard questions to get at, especially in a big recurrent network. And I think ultimately manipulations will be helpful for addressing that.
So I have a question of my own that I'm going to use these last few minutes to ask. There are no other questions from the audience as far as I know. Oh, there is actually a new question. I should be careful. Let me make sure the audience gets their say. I'm going to ask the question I asked on the list here. I'm just reading it for the first time. What are the computational as well as evolutionary benefits of distribution of any function in several areas? And there's a leading follow-up, can compensation after a lesion be related to this? So what's the value of this computationally and evolutionarily? It's distributed.
I think there's lots of, I think what it is in terms of like showing it with data, I think it remains to be seen. I can speculate, right? I mean, maybe having some redundancy for lesion is one possibility. I think having the distributed information is helpful, as I was suggesting, for maybe forming conjunctions of different types of things. And then also when we think about, navigation as this like closed loop behavior where you have to have a lot of feedback, right? It makes sense that all this information has to necessarily be kind of spread around, because you need, you have to interpret some features in the context of others and so on. So it kind of, I don't think would work or be sensible for it to all be kind of separated in very modular ways where it doesn't look to be distributed. So I think that's a maybe a big component of it, at least how we're thinking of it, for having these areas interact in kind of this recurrent feedback type of way to, and also leading to the integration of variables.
I agree. It's quite a hard question. But so the question I have to ask right now is, again, a broad question. You're showing there's some degree of localization, functional specialization that's localized in the ways you've shown. And then there's also this distributed integration that's also going on. And the way the way I think about this often is that there are signals that are being computed locally due to the dense local connectivity of microcircuits, for example, that could then be sent to other areas through long-range projections or, along those lines. In your data, I don't really see that so much. So I don't necessarily see, although you didn't explicitly show this, and of course, maybe it's there, the relationship between where the codes are in different places and the projection systems or the connections that are anatomical that could support the interactions that are leading to distributed.
Yeah.
How do you think about how do you think about this sending of signals or communication question?
Yeah, we've looked. Yeah, so I didn't talk about the anatomy. I mean, these areas are all interconnected, either monosynaptically or 2 synapses away. But we've looked a little bit at labeling cells that project to known targets and seeing if cells have completely populations of cells that project to area A versus cells that project to area B. Do they have completely distinct or completely similar types of activity patterns? And what we find is it's neither of those. It's somewhere in between. There's statistical differences or statistical biases in the types of information that cells might have if they project to A versus B, but there's a tremendous amount of overlap as well. So it doesn't look like it's breaking down by just like separate information routing to different areas. But to zoom out maybe more speculatively, I liked the way you phrased things at the first part of the question a lot, where you said the local circuits can compute things and then maybe send them out to other areas. And that's a lot of how we're thinking about it, that representations can be distributed, but that doesn't mean that computation is necessarily distributed. And this idea of trying to separate out representation from computation is a very interesting and important one. That's part of why we're trying to think about, with 2-photon optogenetics, ways in which we can actually probe local computations within circuits, so we can try to see if we might be able to understand the more local computations that are present in one area versus another. And even though their representations might be highly overlapping, perhaps their computations are more cleanly separable. I think really totally dissociating representation and computation from experimental measurements is a very challenging thing to do. And I think we need better, thinking on the computational end, but also on, experimental tool end to really separate those things.
Perhaps I could suggest and get your thoughts on, do you think, therefore, that the choice variables are relatively weakly encoded individual neurons, as you showed, even in the retrospedial cortex, for example, reflects how this isn't? I mean, I think you've already said this essentially, but I want to draw this out as maybe even a concluding thought, that these variables are not computed locally, even though they're more preferentially represented in certain regions. Unlike perhaps the information that's extracted from sensory inputs or that used to control motor programs. How far do you go along with that kind of suggestion? And
I think it's an open question. I don't think our data can speak to that at the moment. So I think it would all be speculative. But I think it could fall in either direction. It could be that it's computed more in a more distributed fashion, right? As you're suggesting, but I don't think it necessarily has to be that way, right? And I think, at least in our system, the right experiment to pull those apart, I'm not even sure yet we know what that right experiment is, but we haven't done one, I think, that can fully pull those apart.
All right, well, I think that's a very fair answer. We don't have any other questions from the audience, but we still have 188 people watching. So I think at this point we get to say thank you.
Yeah, thank you. Thank you, the audience. Also, thank you, Bijan.
No one else gets to say anything but me, apparently, because we don't have a front-end interface to the audience, so I can hear a resounding round of applause.
Yeah, thank you, Bijan.